but it's a lovely slide, by the way. No, it's wonderful to be here. So I'm very much pleased to be invited. And I first want a small applause for these three lovely ladies. Because I still remember that they want to speak with me, and we had this phone call, and you were so modest. For half an hour, I guess. I think, and then I mentioned maybe get a Bert de Vries Foundation, because they were 10 years sort of ahead, and that worked out very well. Okay, so it's my pleasure to tell you a little bit more about how we did get to the Janssen-de Vries syndrome, Janssen-de Vries syndrome, JdVS, you can call it anyway, doesn't really matter. And I would like to give you some minor glimpse in the future, and the future is already now. I will show you. Okay, so my name is Bert de Vries. I'm from Nijmegen. For you to know, Nijmegen is actually the oldest town in the Netherlands, and it has been founded by the Romans more than 2,000 years ago. So might you be in the neighborhood, I can recommend visiting the city. And let me see, is there a pointer as well? Yes? No, okay. You see this, oh yes, there's a pointer. No, oh yeah, there it is. You see, so this is the Netherlands, and Nijmegen is actually a little bit at the eastern part, so close to the German border. As you can see, the Netherlands, it looks pretty large on here, but it's 250 miles from here up to there. So, well, it's huge, isn't it? But, oh, let me see. Oh, it doesn't do it. Now it has to do a trick, because it will should move into North Carolina, but this is lost, and it fits 4 times in North Carolina. I worked hard on this. So the Netherlands moves and then disappears in North Carolina. Impression how small it is. But with the transition, this is working. Anyway, so it's small for your standards, and we can do with it. So when we're talking about history, I want to start with real history, and then I would acknowledge Antonie van Leeuwenhoek. By the way, he was a Dutchman as well, but he lived in Delft, so that's another region of the Netherlands. He was renowned about his microscopes. So the Dutch microscope is famous microscope. He was very good in making these, and he started looking at lots of things, all things. But he also decided to look in sperm. And when he looked in sperm, he saw the tail and the head, and he started thinking, and he thought, still there? Yeah, that's interesting. Because maybe in this head of the sperm, there might be a little boy or little girl sitting, transferred from the man to the fruitful womb, and then grow out in a lovely baby boy or girl. Interesting thought at this time. It was a considerable normal thought. We know better now. We know better now because since the early 50s, we are aware of chromosomes. Chromosomes that are in each cell. We are built of cells, and in those cells we have chromosomes. And when you look at them, then there appears to be 23 pairs of chromosomes. And they are numbered, as you can see over here, from 1 till 22. And then we have the 23rd pair of chromosomes. There are either that are sex chromosomes, there are 2 X's, you're female, XY, and you're male. Chromosome analysis in children with Janssen-de Vries syndrome, you won't see any abnormalities. It looks completely normal. So we need further steps to get there. So this is a male. So from 2000 on, we developed DNA chips to look more accurate into those chromosomes with a better resolution, even better and better and better over the years. But the major step, and most of you are probably familiar with this, was the whole exome sequencing, so that you can test all the genes in one go, 20,000 genes. So that's amazing. And it looks simple. You have your DNA, you put it into a machine, and the sequence is coming out. I always joke with the people in the lab that it's easy, you put it in and you get it out. But of course, it's not that easy. But then you get all the sequences, so you get all the DNA sequences, and that's huge, 3 billion base pairs. And they all have variants. And then you can test what you want, but then you wonder which variant is actually relevant. So which change in all those billions of base pairs is relevant. And that's a very tricky, difficult thing. So this was developed in 2010. And our group in Nijmegen, we thought, okay, this is interesting. So what we decided to do, and here we were the first one, read my lips, worldwide to use this approach. We decided, okay, so if we test patients, we call it the patient, so don't worry, but if we test the patient with the intellectual disability or the developmental delay, and then we start, what will happen if we compare them with the parents? So something that changed from the parents to the child, and then you can look for de novo variants, the things that happened new. So then you get rid of lots of variants that are just inherited. And we decided to do this in 10 patients. Why 10? It was hugely expensive. So for each patient that we needed to test, it cost 10. So that's expensive. So we use our whole research budget for a year, and we did it. And we're so pleased that from the 10 trios, as we call it, it's called a trio approach, we were able to identify, we could diagnose 7 out of 10 children. 7, that's huge. So we're probably very lucky in selecting the right patients. Fair enough, but so it hasn't been repeated. Nowadays, you pick up around 25–30%. So, okay, but so we published this in Nature Genetics. It's one of the top journals in our field. I said, okay, this is the way forward. And it appears to be the way forward because people are doing it now worldwide. And it's not 10K anymore, it's far more cheaper nowadays. Okay. Where does the story of Bert de Vries syndrome started for me? It was lovely to hear Cindy Curry talk about where it started for her. For me, it started with this 5-year-old girl. And she was referred to our clinics because she had neurodevelopmental delay and some behavioral problems. And the parents wonder, and also the referral doctor, what's the cause? She was born after a normal pregnancy, a little bit low birth weight, and she had some feeding problems as a neonate. Reflux, hypertension, the common things, and probably you will recognize some of them. Her IQ was tested, and she had an IQ of 72, so that's the low normal range. She had some behavioral problems, was very open in communication, and what was striking is that the parents mentioned that she had a high brain threshold. So no problems there. If she hit something, no problems. On the other hand, she was sensitive for sounds. So the sounds was a little bit contradictory. But anyway, so we took the medical history. We looked at her. She was relatively short for her age, but had a normal head circumference. And as a clinical geneticist, you look at the face, and then you can describe the face in different ways. What was striking in her was a little bit prominent forehead, also the shape of the mouth, with relatively broad and thin upper lip and low-set ears and some other minor things. And she walked a little bit unstable, so broad-based gait. Yeah, I cannot do this here on stage. Maybe I fall off then. Okay. So in her, we did this trio approach. So we tested her. So that was at that time sort of routine in Nijmegen and not so expensive anymore. And we identified this, de novo variant in PP1 and D1D. Okay. So we thought, okay, that's nice, but we haven't seen this before. What does it mean? So what we did do is we went back to our over 800 cases we had tested over the past 5 years. And we identified 2 other children, also with a mutation in this gene. And then it becomes interesting. And if you look, then you look at the second one and her again, then I already noted some facial similarities. But then again, if you put 2 children together, there are always similarities. So is this real or just coincidence? We didn't know at that time. Actually, don't worry, I thought this is something new at that time already, but I'm not allowed to say this because that was not very scientific. But I took Sandra Jensen to my office and said, look, there's something new here. We have to go on this. So, and I still remember that moment because I had the photos on my screen. Anyway, so what does this gene do? It encodes for a phosphatase. I won't go into much detail, but it's involved in the DNA damage response pathway. So it's a known gene, but not in the way we are looking at it. And what was striking and for us at first is that the gene consists of 6 parts, and that's are called exons. So remember this, and the variants are always in exon 5 and exon 6. So, and if you look at the gene, then you have here what we call a phosphatase activity. So that's an enzymatic activity that the protein needs to do something in the cell. And here it is this green thing that's called a nuclear localization signal that makes the protein go from the cytosol to the nucleus. So if not there, then it doesn't function very well. And it was striking that we were able to identify 14 patients, and they all had those variants in between those 2 important spots. And both, if you look, they led to a stop. So what the protein is made, but it's shorter. So it's what we call a truncated protein. So you have 2 copies, one normal copy doing the thing what he or she should do, and the other one delivering a shorter truncated protein. Okay, we haven't seen this before, and so specific that all those patients were in this area. And I think this is very important, that's why I pay attention to it. So I don't know what mutations elsewhere in the gene is doing, but it's not causing, to my perception, with the current knowledge, Jensen-de Vries syndrome. So it's not the mutation in PP1D, no, it's the mutation at that right spot at PP1D what's causing Cohen-de Vries syndrome. Jensen-de Vries syndrome, sorry. Sorry for the mistake. Anyway, so what's causing Jensen-de Vries syndrome? Okay, so keep this in mind. I'm not ending anything yet. Can you push a button back there? Yes, thanks. Okay, so then, of course, the exciting moment comes. So you have those 14 patients. That's all done within an international collaboration, by the way. So they are not from the Netherlands, they're from all over the world. So we have our tools to communicate with each other. with scientists all over the world say, okay, we have something, do you also have something? And then you can join. But then, of course, the question comes up, is there what we call a facial gestalt? Is there something we can recognize? And indeed it was. So if you look at the other children, we did see more or less the prominence of the forehead, the shape of the mouth, low-set ears. Yes, there is a recognizable phenotype. And that's also what Cynthia was mentioning. Okay, what was also striking is then they all had relatively short, broad hands and feet as well. Okay, so we noted this and we put it together. And then we start what you do if you have something new, you just look what they all have in common. Now we already knew that the neurodevelopmental problems were there. The hypertonia in the majority of cases, this sort of imbalance, so approximately half of them had this imbalance gait walk, sensitive for sounds, and some behavioral problems. And next to this, we those severe feeding difficulties, like and with the vomiting, and Dr. Bols will go into more detail there on the vomiting and the constipation. They were relatively short. This all children, majority had relatively short stature. The small hands and feet already mentioned, and some had this extra curvature in the spine. And there were also some interesting things which we could not, we still not understand probably, but lots of children had this severe periodic illnesses due to recurrent infections, the high pain thresholds noted in the majority of cases, vision problems, and also the abnormality hypoplastic nails. And as you look at this list, 14 cases, nothing is actually 100%. So this, sometimes it falls. Okay, you should realize that nothing is 100%, but it's relatively far more common than you see in a normal population with children with neurodevelopmental delay. Okay, so this is what we published in 2017. We also added some functional studies, important functional studies. I won't go into detail here, but if there are questions, I can address them. And again, as Karen mentioned, so this was done under her PhD project of Sandra Janssen. She's first author, and I'm last author, and finally it received our names. Okay, so 2017. From there on, what is the next step? What you usually do as the next step is try to collect more data and to see what's the full clinical spectrum, what can we expect. And there Cynthia Curry, Dr. Curry I should say, came in. So we started an international collaboration, Cynthia together with Monica Warczyk, and she will be presenting later on her results. So I won't go into this detail. What I can say is we got 40 researchers from 30 different centers all together, giving data, and they were well put together by those two great ladies. So we overhead at a certain moment 37 extra above the 14 you already knew, individuals with Bert de Vries syndrome. And it ended up in this lovely paper that appeared earlier this year. But Monica will tell you more about this. I only want to focus on one thing, and that's there are also familial cases, and I think that's very important to be aware, because you have all genes in a copy. So you can, so if you have the disorder or the syndrome, then you have a normal copy and the copy with the truncated protein. actually, genetically, it's 50% chance to pass it on to your offspring. You can pass on the normal gene and you can pass on the abnormal gene. So that's 50, 50, 5-0. Okay? Yes. Okay. Okay. I promise you to give you a glimpse of the future. And as I said, the future is ready now. These are facial recognition tools. And we have been working on this in Nijmegen already for several years. We also use this, we developed a sort of model, hybrid model, which is already old-fashioned, but at that time it was very novel. Where you can actually, what we did do is we analyze children with several syndromes, but also for Vries syndrome. We had 11 children with Janssen Vries syndrome, and we compared them with sex age match controls, 55, 5 each, also with neurodevelopmental disorder. And if you look at the results, and I won't go into much detail, it's a T-S&E plot. You see the normal controls in red. You see the children with Janssen Vries syndrome cluster. They group together. But I have to be honest, it looks good, but not good enough, because you want them separate to use it as a diagnostic tool. Okay, so 2D works, but not really, not good enough. So that's why we thought, okay, let's try to get it at the next level. And what's after 2? 3. So let's do 3D imaging. I'm pleased that I got two elegant colleagues, young colleagues with me. So what we are planning to do is, and that's also in the program, to do the 3D imaging. So then you get far more information. But not only 3D imaging of the child with Janssen disease, but also from the sibling, the parents, and even grandparents if they are around. Yes, so then we can start analyzing what are the powerful things what we can actually use to make a clinical diagnosis. And I can say I can make the clinical diagnosis, and Dr. Curry can make the clinical diagnosis. But all over the world, there are people who are not so familiar with the disorder, and they can actually, if we develop this in a proper way, they can use this tool to make the clinical diagnosis. Okay, so in the program it said 2 hours, but it's my youthful collaborators. They are sitting full time, almost. Without, you have your pause, your breaks, of course, at the outside of the, and there will be a form. It's approximately 20 minutes per family, depending on the size of the family. It's lovely to do. I scanned myself as well. Now it doesn't do that good, but that's because of me. But anyway, so they will be doing this. There will be a schedule, so you can schedule in and a time slot of 20 minutes, and then be there and have yourself scanned. Why not? Okay, so that's one thing, and that will be very important. Okay, so the other glimpse in the future, and again, this future is already here. But not yet for Janssen de Vries syndrome, is that you can actually grow from skin cells or from blood cells. You can have them transformed. It's high tech, but you can transform them into eye neurons. So what you can do is you can you can make them pluripotent stem cells, and from there on you can tell those cells, don't tell them, but make them do it, to grow into neurons. And that's amazing. We're doing this now for more than 10 years, a little bit more than 10 years in Nijmegen. And what will happen is that those cells neurons are long, and they form a network. And they get in contact with each other. And not only get in contact with each other, but they also start communicating with each other. So you see all interactions. And now we have to do a certain trick. Yes, and yes. And you can measure this on a MIA, multi-electrode plate. And there you see, so they communicate. So the short communication and the burst, so then they communicate all cells all together at once. It looks like a thunderstorm, but it's not. It's simply cells communicating with each other. And this is so amazing. And the other thing is, the amazing thing is that we've done this for several very rare syndromes, also Bert de Vries syndrome, and they have a unique pattern. So it's amazing that you get those cells, and then if you have the eye neurons from syndrome A, they communicate different from syndrome B, syndrome C. You can keep them apart. I think that's amazing. Now the idea behind this is we're not it's not a brain or whatever. But if you see how this sort of communications is going differently, you can try to see how you can correct it towards normal, because we have also normal, so we know they do certain things at a certain time, in a certain time frame. And this will be, for us, the model, eventually, to see whether we can correct this communication. And so you can sort of treat those eye neurons in this dish, and see whether you can correct the miscommunication, as I should say, or different communication, but different from normal at least, by adding certain drugs. It's a huge step to treatment. Don't worry. So I'm not here. So treatment is somewhere over there, but it's a very important step, and we are making those steps. Not only doing this through this, so I mentioned you have those spikes and those bursts, and so this here you see the neurons lying, and these are the electrodes. Anyway, what you're also doing is you can also make them grow into cerebral organoids, and that's a very popular name, brain in a dish. Again, this is not a brain whatsoever. But it's an initial stage where we can actually study the way the brain develops at a very early stage. And we know how this should be in a normal situation. You always do those. This is high tech. I'm not doing it myself. I have a very good bioscientist with 3 thumbs up and has a team of 20 younger colleagues working around him, predominantly females. They are very good in this. Anyway, we know now we can make those mini brains. We can see what's normal, and normal I mean average. That's how we should define this. So what's average? And then we can now study, and we're doing this for certain syndromes. And I hope to be able to do this also for Bert de Vries syndrome. The sooner the better, in my opinion. And you can see, you can study, and you can see what's going normal. And another thing. Okay, so now we have some knowledge about individuals with Bert de Vries syndrome. What we did do is we created a website, and you can note this thing down there, humangenes.nl/ppm1d, and there you can find a lot of information. The idea we have is part of a website series. Now you recognize some of the names. Probably it's not visible at the back, but Cynthia is also involved, and Sandra Jansen, of course, myself. The idea is this is a website where both professionals as well as families can get information. It's, but another important thing is that we are collecting through this website data from professionals. So your doctor can, a doctor with who had a child with Jensen-de Vries syndrome can actually add the clinical data to this website. Then you have to push a button on the professionals, which is relatively logic, and then you get an upload clinical form. It's all automatically, and this doctor, he or she has to fill in this form in 10 minutes time. 15 minutes if he or she is a little bit slow. But if you have all your clinical data, you can just fill it in. The moment the doctor fills it in, then the message goes to the moderators, it's either Cynthia or me, and we will check whether this is a real doctor and if it's something real, and if it's well. So we create the data. And when we say this is okay, then it will be added to the graph and chart. And this might be very important for you when you go to your doctor. Because here are all the data currently known, at least known to me. I know there are far more patients than those. I think we're currently on 55 or something children added. By the way, if you have been part of the study, you're already in there. Because Cynthia and I worked on this. Yeah, so then you don't have to, then you're already there. But it's for the new one, so that a doctor from Adelaide, for instance, who has a child with Jensen-de Vries syndrome can say, okay, it's important, I want to share the knowledge, what we do have, to the community. So this is one way to collect this data. And that's very important, because if something happens only, a specific feature only in one child in 55, it might be coincidence. But if you have 3 or 4 or 5, then it might be thinking, okay, we should be aware of that it might be actually linked to the syndrome. We are currently working, and that's also a very important view, we are currently also working on a parental portal. That means that the parents can actually add their data of the child, or even the children themselves. You can sit together. That won't be done in 10, 15 minutes, because if we open this, we will ask you lots of questions, a long list of questions. But that's very important, because I don't know anything about Jensen-de Vries syndrome. I have to get the knowledge from you, and then I can put them together, and then we all know the same thing. This is what, on this website, is what we know about Jensen-de Vries of 55 children and young adults. This is what we know. I don't know more than you do, so it's completely open. And with the parental portal, and that's probably will be opened, I still have 10 minutes, but I'm already closing, don't worry. Then we will be able to create a natural history. Because we do want to know when certain things start and when certain things happen, when certain things, for instance, the cycling vomiting, you want to know what's the age on average it starts, and is there a difference between individuals? Does it go on? And you might also learn about certain treatments. Okay, conclusions. I'm in time there. It's a new syndrome. It's very new, so we don't know much, but we learn a lot. But the only way we learn from you. You're not from me. The only thing I can do is, with our team, collect data. Keep in mind, exon 5, exon 6. If it's not there in that region, then it's not, to my opinion, at the current knowledge, not Jensen-de Vries syndrome. It might give other problems, I don't know, but we don't have those data. Yes, so keep this in mind. Okay, this is what I briefly mentioned. So there are now 55 individuals with Jensen-de Vries syndrome in this database, and we will be keeping collecting on, and we'll probably, and that can be entered from all over the world. What do we need to do? Thousands of things, but I only mentioned 3. We have to get good clinical data in. We have to get more knowledge about the natural history. I discussed last night with a couple of members here in the audience, parents, and said, so we're now at position A, and you have thousands of questions. If we don't start working on this, then in 2 years' time, you have still the same questions. So we have to move from A to B to C to D and get there. But, okay. I want to improve clinical diagnosis. I want to make things easier for clinicians out there who are not familiar with the disorder. 3D imaging might be a way forward to do this. Okay. And of course, I think we should try to get things at the next level and also do the eye neuron research and the brain organoids, also for Jensen de Vries syndrome. Okay, having said this, I would thank all the professionals that collaborated to get all these data together, a huge team in Nijmegen working on this. I'm pleased to work with them, but of course it's all about you.